All right, so on behalf of the Pancreatlas team, I'd like to start by thanking dkNET for inviting us to present today, and to all of you who have logged in to learn more about us. My name is Diane Saunders, and I'm joined by a few colleagues who you just heard introduced. And briefly today, our presentation is going to be divided into 4 main sections. So first, I'll give you a quick background and rationale for creating Pancreatlas. Then we'll dive into the technical side of things, give you a demo of the data that's available, and then wrap up with some future plans and hopefully input from all of you attending. You heard from Glencoe that you can put your questions into the chat, or we'll have plenty of time for discussion at the end. One of the things that I think makes Pancreatlas such a unique resource is that it was created and developed by a multidisciplinary team. You just heard about myself, JP Cartailler, and Marcela Brissova. So I have a biology background and an interest in design. JP is a bioinformatician and a database developer, and Marcela is a leader in human islet and diabetes research. And so having developers and biologists in the same room from the very beginning of Pancreatlas's creation has, in our opinion, been instrumental to our success. And I think you'll get a sense of that as you hear from both JP and Marcela later on. Many of you are probably already familiar with the pancreas, but for those who aren't, the pancreas is a bifunctional organ that is part of both the digestive and endocrine systems, situated in the body between the liver, stomach, and intestines. The vast majority of the mass is composed of acinar cells, which produce digestive enzymes, and they're secreted into a branch ductal network that empties into the duodenum. And then scattered throughout the pancreas are islets of Langerhans, which are composed of hormone-secreting endocrine cells that regulate blood glucose. So given its bifunctional nature, there are quite a diversity of diseases in the pancreas that can be quite varied. But these include diabetes, chronic pancreatitis, pancreatic cancer, and cystic fibrosis. And several funding agencies, including the NIDDK, have really started to fund research programs that are aimed towards identifying both causes and treatments for these conditions. In the past decade or so, rapid technology development, as well as the increased availability of islets and human tissue for research, has led to the establishment of a number of different programs, some of which are shown on the screen here, aimed at characterizing really in-depth the human pancreas. So some of these, like NPOD, the Network for Pancreatic Organ Donors with Diabetes, is obviously focused around a disease state, but others are focused more on development, such as the Handle program, or characterizing and creating a reference of an organ over the lifespan, such as HubMap. And since the pancreas is not easily biopsied, human pancreas research really relies heavily on accessing tissue from deceased organ donors. And NPOD has done a tremendous amount of work to establish partnerships with a network of organ procurement organizations, or OPOs, throughout the US. And this infrastructure is what ensures that pancreatic researchers have access to high quality organs from individuals of diverse backgrounds. Obviously, many different types of data are being generated from pancreatic tissue, and these include functional characterization, various omic profiling, but our phenotyping efforts here at Vanderbilt, along with several of the other programs that I've mentioned, have focused really on acquiring cross-sectional imaging data to better understand both tissue architecture and spatial relationships between cells. What this means in practice is analyzing hundreds of tissue blocks in various formats, generating thousands of images in different modalities. These include confocal microscopy, traditional immunofluorescence and immunohistochemistry, whole slide scans, and multiplexed immunohistochemistry. A few years back, we began thinking about how to compile all of this data in a meaningful way that would link the de-identified clinical information along with the high resolution images and data acquired from there, and do all of this in a way that would be easily accessible to the rest of the research community. We started off with data from the 4 programs that are listed here, and we conceived of an atlas, which eventually became named Pancreatlas, that would serve as an imaging resource detailing the human pancreas in both health and disease. Our first task was to address some of the challenges I just mentioned. For one, there are tons of images being generated, so we wanted to make sure that that data was easily explorable and accessible to those that were new to the field. We also knew that there were a lot of different file types. New ones are being generated and developed every month or two, it seems. So we wanted to make sure that there was support for common file types, but also for the more proprietary image types that require their own separate browsers oftentimes to access. As I mentioned, because these samples come from human tissue, we wanted to make sure we captured and kept associated metadata right alongside the imaging data to better inform studies. And also we knew that traditionally images are published as a small static panel in a figure. And so one of our priorities was being able to provide a platform that would enable users to interact with images and also would support some of these more novel imaging technologies that have 10 or 20 or 30 different channels. So to explain how we addressed some of these challenges and kind of achieved our goals, I'm going to hand it off to JP.
Okay, well, thank you very much, Diane. So indeed, I'm going just to continue these slides, just going through some of the technical background of how we've constructed Pancreatlas. And I'm happy to take any questions at the end of the talk. Okay, so just to add to what Diane was just speaking about, sort of our requirements was we really had a balancing act to figure out from the very beginning of this project. We really wanted to balance usability and complexity. It's really not a task that's easy to achieve because, first of all, we wanted to provide a platform that was going to be relatively easy, if not very easy, for our primary audience who are biologists. But also we wanted to have enough complexity that it would be possible to access the data, not just through this interface by biologists, but also by potentially developers and analysts who might want to do a lot more complex tasks with that data. So with that said, in addition to priorities revolving usability and accessibility, as Diane detailed earlier, we really did not want to reinvent the wheel. Personally, one of my mantras is whenever we have an idea is just to go out there and start researching what other people are doing and to see if we can leverage existing methods or technologies. We obviously wanted user-friendly online tools, but really one of the key aspects of that was to not require people to download or install anything. As you may be aware of, is if you want to access some of these imaging data sets, I mean, even sometimes a single image can be 20 or 30 gigabytes in size, or at least several gigabytes. So it's nothing you can interact with from a website, even if you have access to the data. You would need to download it, install software, configure the software, hope that it can actually read that file format, so on and so forth. In short, it's a lot of friction. And with that said, we wanted to support all image formats, not just current image formats that are commonly used by different manufacturers and platforms, but also future formats that are still either in development or have yet to be invented. And we didn't want to have to do that ourselves. That's just much too much work. And as much as can be supported by the funding available, we also wanted the platform to be scalable and, again, flexible. So we, after spending several months, Marcela, Diane, a few others, and myself, talking to different vendors and looking at what was out there, we opted to use Omero Plus. So Omero Plus is a commercial version of the open source Omero software. It's packaged and further developed by Glencoe software. And it's really just it's an excellent platform for image management analysis. And basically for us, it was about, we didn't want to get into the business of creating these platforms. We wanted to get into the business of building a Pancreas atlas. So by working with Glencoe software, it really became a turnkey solution where they could essentially install this platform onto our servers, which we did within the context of a private network at Vanderbilt University Medical Center. So the software was installed locally in our IT infrastructure, and over time it was connected to LIMS and image storage as well. And then we got into the task of actually working on our custom web interface, which was a de novo project, which would also connect I'm sorry, it's not moving forward. Thank you. It would then connect to PathViewer, which is also another component that's provided by Glencoe software. PathViewer is essentially it's a microscopy image viewer. I think it was originally developed more for pathologists, but one of the key features of it is it can display more than 4 channels at a time. So ultimately, really, we wanted users to have the ability to access an interactive and full-featured image viewer, which is possible through PathViewer. And at the time that we made these decisions, that was really the only option out there. Since there's a few open source options which are currently under development, which are also very promising. So we're really excited to see that. I think it's just good for the community to have more than one option. And we, especially through a lot of curation efforts, we were able to develop curated points of entry for each of our data collections, so that way people can access these large arrays of images in a context of disease or phenotyping efforts, so on and so forth. And again, by leveraging Omero Plus, we could access all these images. And just so you know, it comes with this quote-unquote bioimage library. It supports over 150 file formats, including some of the newer formats of data that we get from, say, Codex imaging, as well as the up-and-coming OME-ZAR format, which is an H5D derivative. So pretty excited to see that, because that's going to really open the gates for a variety of new types of data to be embedded into the actual images as well, especially as we go into multimodal types of efforts. And in short, this was really a key thing for us. But with that said, Omero, for those of you who may have used it, has a web interface that comes out of the box. We found that it really did not provide a good user experience. I think it's a fine system. It allows you to access your collections of images, do some simple sorting and filtering. We really wanted something that was more modern looking and just much faster and more interactive and didn't require page refreshes, so on and so forth. But we love the PathViewer application for actually looking at the and interacting with the imaging data. So we decided to, whoops, wrong way. So we built some layers in between these two. And the first layer we built was a backend API that we built through Django, which is a Python web framework. And then on top of that, we built a user interface using React, which is a very popular JavaScript library for building user interfaces. And the user interface was really built for biologists. So our focus was really 100% of end users. We want people to access the website and just know how to use it without having to be trained. We used really common web idioms, so we wouldn't be reinventing the wheel. And more importantly, we didn't want to train anybody. And React also has a very large community, so there's lots of places to go for ideas, as well as support when we get stuck with something at a technical level, for example. And Django is really more for analysts and developers. This is more the sort of machine-friendly interface. This is where we do a lot of the metadata as well as image retrieval directly from Omero, then pass it off to React. We also have stubs in place from the very beginning for non-Omero data. So very early on, we knew that we wanted to do more than just imaging. So we essentially built all the stubs in place, and we're now coming back to start to put in non-imaging data into Pancreatlas, and we'll talk about that later on. And this also will be the place for a future public-facing API. And as we were wrapping sort of our first version of Pancreatlas, we decided to open source our work. So we took Pancreatlas and we forked it into a data agnostic version. So we kind of stripped everything that was Pancreas from it. And we ended up packaging it up and publishing on GitHub. And if you actually go to GitHub and you're technically oriented, you can actually very easily clone the repository to your local machine. And it really only takes 2 minutes to get the application up and running from the time you actually clone it to the time you actually have an app running in your web browser on your local machine. The FFIND repository also provides you with flat file data sets. So these are all mocked up data sets. So they're very easy to go in and tinker with and essentially reload the page and you'll see your changes. Then you can essentially substitute those for API endpoints if you're interested in passing up some real data through these JSON-based interfaces. So we've put the URL down there. I think it's going to be in the chat window as well. So we encourage you to check that out. If you have any feedback, please just post an issue directly in GitHub and we'll get back to you on that. So that's kind of our technology stack. So now how do we actually get data into it? So again, we knew that the people who would be providing the information would be biologists. So we wanted to have also as easy of a way to get data in. So we worked with the Glencoe software developers to put in place an appropriate import scheme. So we could only, people such as Diane could essentially generate a spreadsheet without involving any developers for the most part, except for the time of loading, and be able to essentially fully annotate these spreadsheets where we have all these researcher-defined variables that go as column headers that we can obviously, through Excel or through another system, sort of enforce, the control vocabularies, things of that nature, and do that for each of the images. And then as that spreadsheet gets finalized and all the images are put in the right places in our data storage, we can essentially import the spreadsheet, and then the Miro backend will go ahead and process all those images and create all the image pyramids as necessary and store all that information, and essentially then build up the metadata catalog as well as the key value store that our Django backend will essentially use to feed Pancreatlas data with. At that point, it's simply a matter of us publishing the data stuff in Miro, and then it pops up in Pancreatlas, and we can just share the link with the world. So since inception, we've put in 7 collections. Again, these are the curated points of entries that we mentioned earlier. We're currently working on a new collection that comes from Exeter. And, as you can see here, about half and half. Half were from phenotyping program efforts, and the other half were from manuscripts. And just to highlight what Diane pointed out earlier, you publish a paper, you may have some imaging data, you have a couple figures, and you can only zoom in so far, and they're not interactive. But you may have collected several dozen or maybe several hundred images. We're just putting these in as collections that are associated with manuscripts, so people can further dive into the data. In terms of growth, since version 1, our first release of Pancreatlas contained nearly 400 images, and this was mainly the Handle P, the program at the time, and now we have nearly 2,000 images. We also monitor usage. We use Google Analytics. Caveat of that is if you have through your web browser, if you've installed things like uBlock or third-party tracker prevention software like I do, you won't be monitored. So these are certainly incomplete, but we really use it more just to look at trends. What was really cool is when we published our manuscript in bioRxiv, we actually got a very significant peak. Whereas when we actually published the peer-reviewed manuscript and patterns, we really didn't get any impact. So it just goes to show that, at least in this anecdotal evidence, bioRxiv is definitely a good way for us to communicate the existence of Pancreatlas to the world. Well, we have seen those in the last year. We've definitely seen an increase in usage, and we hope to continue doing that by doing, getting the word out through conferences and webinars such as this. And of course, user engagement still is important to us. And with that said, I'm going to pass it back to Diane for the site tour. Thank you.
Awesome. Okay, so as you heard from JP, as we built Pancreatlas, we really tried to continuously think about what gaps there were in the field, both in terms of tools and the biology. And so hopefully the list that I'm about to show you is going to grow as we partner with more investigators and highlight different aspects of pancreas biology. But here are just a few examples of topics that are available for exploration right now. So the first is related to our Handle P collection, and that is, how does the architecture of the human pancreas change during the first decade of life? So we've got hundreds of images that actually really beautifully illustrate all of the dynamic changes that occur. You can look at the entire cross-section of a pancreas and appreciate the changes in density of endocrine cells, the rapid expansion of growth of the pancreas. And so I would encourage you to check that one out if you are interested in development. Another thing that we have started to showcase are the cell and tissue level alterations that occur in individuals with type 1 and type 2 diabetes. So there are collections through the Human Pancreas Analysis Program, as well as a couple of different manuscripts that will provide kind of imaging data that comes from both non-diabetic donors as well as age-matched controls along with individuals with type 1 or type 2 diabetes. So that can be really useful as you start to appreciate some of the differences either in marker expression or overall pathology. One of the things that we've become interested in recently through our research group's involvement in HubMap is really understanding how much variation exists across the histological features of a clinically normal pancreas. And there have been ongoing discussions in the field about kind of what constitutes normal. So I think that one of the benefits here is that we have a pancreas from a lot of different sources. We have a lot of different imaging modalities. And so I think it really provides the opportunity for those who are, maybe new to the field or those who are starting to think about specific studies to really understand kind of the range of what normal is and what the tissue looks like in cases of various ages. And then finally, we are interested in kind of building up a panel and information about markers that can be used to visualize specifically in images, various different parts of the pancreas. So including exocrine and ductal cells, stromal and immune cells, cells that maybe are less studied in terms of they're not islet cells, but they have very important implications for diseases like pancreatic cancer and chronic pancreatitis. So today I'm going to use this final question to kind of dive into the resource itself. To give you a quick tour of the website, pancreatlas.org. From the homepage, you can either click image collections or the down arrow to view all data sets currently available. If you'd like more context, select collection details to read more about the project, or you can simply choose a collection and begin browsing. Once in a collection, you'll see a grid of available images, each of which has its own card that lists important donor and sample characteristics, as well as the different markers visualized. On the left here, you see metadata filters, which will refactor automatically to give you available images as you select different attributes. Clicking on the magnifying glass launches a modal preview, which enlarges the image thumbnail, gives some additional sample information, and then allows you to navigate directly into PathViewer, which is Glencoe's web client for exploring images. So this particular image was acquired using Codex technology, which is a fluorescent immunohistochemistry-based multiplex system. This is a cross-section from a non-diabetic adult donor, which is full resolution at 20x magnification and contains 28 registered channels. So notice that the image is loading almost instantly despite being more than 25 gigabytes, meaning that you can immediately browse and interact with multiple images within a given data set without the friction of long download times. And instead, PathViewer really lets you dive straight into the data. So first, I want to just briefly highlight the list of markers to the left. These are grouped into biologically relevant categories. So not only can I pan around the image and explore areas of interest, I can also toggle on and off channels depending on the cell type that I'm interested in. So for example, right now, the endocrine cells, hormone-producing cells are shown, but if I wanted to, I could add in a vascular marker to visualize islet capillaries, or I could choose an immune marker in order to understand the proximity of macrophages to various structures within the pancreas. You can see there's ducts highlighted here in cytokeratin in orange, and then you can also see the ecadherin that is marking individual acini. The flexibility of PathViewer also means that in the future, we'll be able to import dynamic object level masks like cell type or cell neighborhood annotation, which can provide even more context to the raw images. A couple of other things I just want to briefly mention. From the header menu, you can easily access our nomenclature tab, where we've provided accessible descriptions of terminology and have tried to really standardize metadata across our collections. And this can be particularly useful if you're looking for markers to label a specific cell population in the pancreas. And we've even linked to the Human Protein Atlas in pertinent cases so that you can get even more comprehensive information. In addition, we hope that our resources page kind of acts as a one-stop shop for other resources. This is particularly designed for trainees or investigators who may be new to pancreas biology and curious about what is out there. Okay, so I hope that kind of piques your interest and you go explore for yourself later on. But for right now, I want to invite Marcela to share some of our visions for where the resource is headed next.
Thank you, Diane. Let's see. All right, so as we are looking forward on the side of technical advances, we would like to build knowledge graph, improve compliance with FAIR principles, and also add new annotations. This will be coordinated with a common coordinate framework that is being now developed by our HubMap colleagues. On the feature-based development, I would like us to focus on these three areas. So as you heard from JP and Diane earlier, Pancreatlas currently is housing 7 different collections, and as these collections are growing, we would really like to enhance search and browsing features within the Pancreatlas so that you can view all data sets in this one single grid, and also be able to search and filter across collections to identify data of interest. And we would also like to improve a layout of donor and sample filters here. Now, I feel the power of ongoing pancreas mapping efforts is really in multidisciplinary integrative approaches, starting with imaging data sets where one can really define spatial relationships at single cell level and overall tissue architecture, and then overlay this with cellular physiology, adding single cell transcriptome and chromatin signatures, and connecting it all the way to genetic risk and variants. And for example, in the diabetes area, these multidisciplinary integrative approaches are now paving the way to cellular attribution of GWAS loci and signals to inform the mechanisms underlying personalized signatures of genetic predisposition. Now, this slide is illustrating how we are envisioning to connect imaging and omic resources. So what we would like to do on imaging side is this specialized path viewer that can allow for direct interaction of users with images, and then add specialized viewer for our omics data, where you can see these omics data would be populated using these cards as we are using for images in the Pancreatlas. Now, our next big step is going to be uplift of Pancreatlas into the cloud, which will provide really increased scalability for this platform, and also simplify overall data upload for external collaborators. Importantly, it will be easier to access raw imaging data, and one can also run image analysis directly in the cloud. As you heard earlier from JP and Diane, Pancreatlas was built as a connectivity platform with its FFIND feature. And currently, Pancreatlas is connecting 4 major pancreas mapping efforts here in the United States. But we are hoping that in the near future, Pancreatlas will be able to connect other initiatives focused on the pancreas mapping, such as, for example, in Europe, SPAS program that is supported through Human Cell Atlas Pancreas Initiative, or HubMap program. We would also love to reach out to scientists in the area of chronic pancreatitis and pancreatic cancer. So it is really exciting to see what the future may hold. This brings me to acknowledgement. First and foremost, I would like to acknowledge our Pancreatlas team. It's a wonderful group of talented individuals that I had a great privilege and honor to be part of. I would also like to say big thank you to our collaborators at NPOD, Handle, and HubMap programs who are primarily based at University of Florida. Big thank you to our contributors to Pancreatlas. And I would also like to thank our funding agencies supporting this initiative, especially to Humbly Charitable Trust. Finally, I would like to express our deepest gratitude to organ donors and families without whom none of this would be possible. And with that, I'm gonna now invite you to, if you could please complete this community survey. It's a really 3-minute survey through menti.com. Here is the code for it and also QR code. And you can do this either now or during our Q&A session. Also, we would love to connect with you. We would like to hear your feedback and suggestions. Here is the email address where you could send those. And if you would like to upload imaging data sets to Pancreatlas, please reach out to Diane. Here's her email address. And if you'd like what you saw and would like to join our team, we are hiring for the positions of a graph database developer, data curator, and project manager. So please reach out. And with all of this, I'm going to turn it over to Koei to lead our Q&A session. And I see a lot of questions popping up in the chat. So hopefully this will be fun. Thank you.
Yeah, thank you for the presentation. So now is our Q&A session. Let me check. There's several questions in the chat. Some of them already answered, but I'm gonna repeat the question and then see if other people missed this question and the answers. So the first questions come from the Mirada. How programmable is the platform? Can developers do tool building? For example, building specialized image processing method using the data on the Pancreatlas. Yeah, I think JP already answered in the
chat. I can definitely just kind of add to it. I was trying to type as fast as I could, but try to get as dense as possible. I mean, sure, yes. Firstly, the Omero system has language bindings. You can interact with Omero, which means interacting with the image data directly and the metadata directly in a programmatic fashion. The way most people do it is they have an analysis server that sits, it has a different machine, so you're not clogging up Omero's environment by trying to do analysis. So yeah, that's another advantage of using the OME or Omero platform in general, is because all this stuff's already been hashed out because there's thousands of people using it and they all want to do analysis.
Okay, thank you. And then second question, how is the information on the image pre-processing done? For example, removal of artifact per filtering. How annotated, curated are the image data? Is information available on things like how field of view was selected, scale information, etc.?
Yeah, this is a fantastic question and something that we've been thinking about a lot. And so it really depends on the data, and I think, or on the collection, I should say, and the type of data. So we are definitely working on adding more metadata. I know there's several different groups out there who have done tremendous work to try to standardize some of these really important metadata fields for microscopy. And so that's definitely something that we want to add. In most cases, what you're seeing are unprocessed images. They're just either scanned using a whole slide scanner or imaged, in another format. And so for sure, we want to add more of the kind of image capture metadata. And I think that that's an important step towards, being more transparent and reproducible. So great suggestion. And we hope to deliver on that in the future.
Yeah, and I think next is a comment. So it's about scrolling down over the disease facet. What is the ND means? Is it means no disease? Yeah.
Yeah, so this is actually on the top of my wish list. Since I'm not a developer, I can't promise you that it's going to happen tomorrow. But yes, you did notice that during that demo that if you scroll over the metadata fields, it defines the kind of the category. But where kind of the next step is, yes, to be able to define all those abbreviations just with the hovering. At the moment, you can definitely find them all on the nomenclature page, but I realize that's not an ideal situation, so we're working on it.
Yeah, just to add to that, along with those future goals of, adding those annotations more within the interface so it's readily available and clear and open, we'll also be augmenting this with a lot of ontology terms, or terms are used by other, by established ontologies, which will also make it easier to cross-link to other efforts. So they have a standardized language.
Okay, thank you. The next question, will there be provisions for facilitating building tool exchange for users of the data, e.g. a package code repository?
Well, we have the code repository for the FFIND. So we don't plan on having a quote-unquote package. I think this is just better served as a code repository that other people can fork and adapt to their own needs. With that said, if anybody wants to work with the FFIND application and contribute code back to the code base, we're more than open to doing pull requests. That's great. And I think, let's see, what was the other part of the question? Sorry, I have to scroll back up. Versions. Yeah, I'm not sure exactly. Facilitating build tool exchange for users of the data.
You can unmute yourself rather to add to the, if you would like to, we couldn't hear you. Okay, I see Miranda unmute, but we couldn't hear you. Yeah, so it's fine. You can add to the chat as well. Let's move to the next question from Anita. So can you please describe how you handle provisioning of DOIs and other FAIR criteria? So how would I cite a data set?
Yeah, great question. JP is going to kill me because we promised no live demos, but I'm live on the website right now. And if you actually scroll down to the bottom, we do have a very, very early in development data usage and citation policy. So if you click on that, you'll get to this page. And so we do have an RID thanks to dkNET. And you can, of course, cite the manuscript. And then what I've tried to do here, which we'll be adding to, obviously, is some of the images, if they are associated with the manuscript, already have a DOI listed, and that'll show on the image card and also in Path Viewer as you're exploring. And then for some of these phenotyping efforts, they actually already have their own data citation policies. So that's something that we can potentially start to incorporate kind of closer to the image itself, or at least the image collection. But certainly, I think for some of the cases of publishing for manuscripts, what we've done is provided a URL with essentially it's a collection identifier, and people have provided that to the during their copy editing process in the manuscript. But working on issuing our own DOIs is not something we've done yet, but could potentially be something if this is a popular request that we could explore.
Okay. And the next question is from Michael. Do you plan to incorporate spatial transcriptomic image into the data? Into the atlas, yeah.
Marcela, you want to talk about some other data types?
Yes. Hang on, I got a little lost here. Yes. I mean, we are in phase 1 of adding multi-ohm data sets to Pancreatlas that are going to be focused on primarily pediatric pancreas, and again, we would love to intersect with other programs that are mapping pancreas at transcriptome and chromatin at single cell level, and bring those to the Pancreatlas using, you know, the omics data specific browsers and intersecting those with a browser that we are using for image interaction. And as Diane mentioned earlier, we began the process with Omero to actually start bringing also image analysis data and be able to overlay those over actual images to overall improve the user interaction with the image, also from analytical standpoint.
And I think that the file formats, because usually spatial transcriptomics or sometimes spatial transcriptomics are, you know, starting to get standardized, I think it would definitely be something we can look into. I'm not as familiar with those technologies, but because Omero is such a flexible platform, I think it would certainly be something either we could use an existing like path viewer for, or we could find another a browser or a viewer that would be able to handle that data.
Okay, well, thank you. So there's another question from audience. Can any scientists upload their pancreas image to the Pancreatlas, or do they need to join any membership?
Yeah, sorry, this is something that I think we missed in the presentation that I usually say, and that is that while we are a resource, we're not a repository. So we do want to make sure that the data that's coming into Pancreatlas is both, you know, well documented and annotated, and also that it is of good quality and that will be useful to the community. And so, you know, really because we've built this pretty robust pipeline, we don't require, you know, you to join anything, you know, you don't have to sign into anything to use Pancreatlas. But if you have imaging data that you're interested in sharing of human pancreas, just get in touch with us. I'll maybe drop my email in the chat down here too. But we can definitely talk with you more about, you know, whether it be manuscript or you have a specific kind of disease type or something like that you want to share. Certainly we would love to hear from you. And that's kind of how we started this collaboration with the group at Exeter. So Sarah Richardson and Noel Morgan, you know, they reached out to us and kind of said, you know, they were aware of the resource, and we now have a really great rapport and are hopefully going to get their images published here in the next couple of months.
Thank you. So there are other comments in the chat. There are not question from Mirada said this is an incredible resource. Thanks for the team who made this possible. And there is comment from Heiko. Congratulations to the team to implement this wonderful resource. And from Marianne, very nice resource. dkNET can always available for fair consultation. And then there are other great comments, and then I'm trying to scroll down to see if there's any other questions.
Yes.
Thanks for so from Tanner Jackson, and thank you for the presentation, for such wonderful, powerful resource. I'm curious, when you look at the images can you turn off different stands? For example, if there is an image that contains DAPI in GCG and the SST, could you turn the DAPI so you can just see ILS, GCG, and SST?
Yeah, okay, so I'm gonna, again, JP is hyperventilating. Okay, so like this is an image of a neonatal human pancreas. And yeah, so for a lot of cases like this, you know, you can kind of look at something maybe that's overlaid or colocalized, but then you know, you're squinting at the paper and trying to convince yourself whether or not what you're seeing is really what the authors are telling you that they're seeing. So yes, if you come over here in Path Viewer, you can see I just clicked on this little arrow button here. And so the 3 channels in this image actually are just DAPI, somatostatin, marking delta cells, and Ki67. So here, what I can do either from this menu directly or from these sliders where you can actually control brightness and contrast, you can toggle these on and off. So if I turn off DAPI, you know, you can still appreciate where some of these nuclei are positive for Ki. or if I'm looking at a KI stain, I can turn back on DAPI and make sure that it's actually a nucleus. So yeah, we hope that this is, this is why we selected PathViewer and used Omero Plus and Glencoe. So we'd love for you to go play around with all the different channels.
Okay, thank you. So there's one more question. Are there user tutorial embedded in the viewer?
Yeah, this is an interesting question because we actually don't, since PathViewer is made by Glencoe, we don't actually control most of the kind of visualizations and organizations that you see. So we are in pretty close contact with them in terms of giving them feedback about what our users are saying. And so yeah, that's definitely something we could think about, because I do agree that there are little kind of hidden things that sometimes change with new releases and you don't know where to find a menu. So it's a good idea, a good suggestion that maybe we can get a tutorial up there. I'm not sure we would be able to embed it in the viewer, but maybe we could make it in an obvious location where people could access it. And I also just want to mention too, there's some great open source tools out there. JP alluded to, now there's a whole bunch of different image viewing tools that have been developed by some of these big programs. And there's also open source tools that are kind of a more guided tour of images. So Minerva Story and Minerva Author is a really fantastic tool that was developed by Peter Sorger's group at Harvard. And so that's one thing I want to try to incorporate too. So that's a different viewer, but it would be more of a kind of narrated and curated exploration of an image where you wouldn't necessarily have to know what you were looking for. You would be kind of taken through it. So that would be an alternative. It's not really related to your question, but I wanted to mention it just because I think it'll be a good teaching tool and kind of an add-on to complement to what you can do in PathViewer.
Okay, thank you, Dan. And then, okay, I see JP is answering some.
Yeah, sorry.
Yeah.
There's also a question from Juan. Where'd it go? Yeah, to import the raw images from the website directly. Diane, do you want to take that or shall I?
So are we talking about image import or download? Just to clarify. I think, who is that?
I think it's Ron. Let me see. Juan.
I mean, the fact that it says raw data makes me think that it's download, but I'm not. So that's not something that we have capabilities for right now. And part of that is just that a lot of these images are very large. And so, as Marcela mentioned, one of the reasons why we're pursuing lifting everything into the cloud is that, really systems are becoming developed where different data sets can be stored in different kind of data tiers. And so one of the things that we could envision for the future would be that if you wanted to download a specific, data set after, browsing different images, figuring out what you're interested in, that we would actually be able to kind of build you like a custom data package that then you could either offload from the cloud if you were going to do analyses on it, or if you are already computing in the cloud, if you could, you could just access it directly. JP, I don't know if you want to add to that.
Yeah, just one more thing. I mean, we're still in the early stages of this whole uplift into the cloud, but we've done a lot of work on other projects in the cloud. So what we're hoping to do is, so we're aiming at AWS, essentially Amazon's cloud. We're going to look and try to pick the best zone to go in that would be the most beneficial for that's a very difficult thing to do, but we're probably going to end up in US East 1 or 2, which makes a lot of sense for us. So that means if you actually have, if you do work in the same zones, you should be able to, we want to set it up so you'll be able to access the data at no cost. So, some of the biggest costs on these cloud providers is the egress, or meaning, downloading the data off of it. So we want to minimize that cost on us, but also we don't want to pass it on to you necessarily. So, but stay tuned.